Okay, so now that we've taken a textual look at the structure of arteries, let's actually go ahead and take a look at what they structurally look at. So whenever we take a look at arteries, here's our diagram, the artery here on the left-hand side. The first thing we see is we see this inner layer of simple squamous epithelial cells. These simple squamous epithelial cells that make up the innermost portion of the wall make up are referred to as the endothelium. Next, remember that simple squamous epithelial tissue, all epithelial tissues have this kind of protein base here, kind of a sticky base of protein material that the cells adhere to form into sheets. And this is referred to as the basal lamina. And notice here, while we didn't talk about this in the previous slide, we also have this layer of connective tissue that surrounds the basal lamina. This is referred to as the internal elastic lamina. This is a very important structure right here. While it's very thin, it provides structural support. But at the same time, because it's made up of elastic tissue, it allows the blood vessel to give whenever blood pressure goes up. So if you check your pulse rate, you can feel every time the heart beats, you get that surge of blood going through the vessel, and that surge of blood causes the vessel to distend a little bit. Well, this internal elastic membrane provides structural support so it doesn't rupture, but does help to allow it to distend somewhat. In addition, you may notice that it has these little tiny pores in it. Those pores are very important because they allow for the diffusion of substances through the walls of the artery itself. So the artery is technically an organ, and it's made up of cells, and the cells are going to need to be fed. They're going to have to receive nutrients and water. The internal elastic lamina helps to allow for the diffusion of substances from this internal area where the blood's actually moving, which is actually referred to as the lumen, helps to allow materials to diffuse out of the blood through the layers of the walls to provide the cells with the nutrients and water that they need. Okay, so the next structure we have outside the internal elastic lamina then is the layer of smooth muscle tissue that we talked about. And again, surrounding that layer of smooth muscle tissue, we're going to see another layer of elastic lamina. This time, because this is further toward the external surface or more superficial, it's referred to as the external elastic lamina, but provides more of the same function as the internal elastic lamina. And then finally, we have our loose connective tissue that surrounds and makes up the outer wall of the artery, the areolar connective tissue. So looking back then, the endothelium, the basal lamina, and the internal elastic lamina make up the first layer of the arterial wall, which recall is called the tunica intima. The smooth muscle tissue and the external elastic lamina then make up the middle wall, the tunica media, and the loose connective tissue makes up the outer wall of the blood vessel, which is called the tunica externa, or remember we said it's also called the tunica adventitia. And finally then, I put these two names in. This represents the lumen of the blood vessel. This is the internal cavity of the blood vessel through which the blood actually passes, is referred to as the lumen. Okay, next up, we take a look at arterioles. And if you've had medical terminology, you may have learned suffixes and their importance and how they change the meaning of terms. So here's a suffix, OLES. This is a diminutive suffix. So when you see this attached to a root word like artery, OLES is a diminutive. It means a small version of the same thing. Okay, so OLES is a diminutive ending again, and when we add it to artery, it creates arterioles. Arterioles then are small arteries. Um, these small arteries, the arterioles, are just small enough, or just big enough, I guess I should say, to be visible to the naked eye. Whenever we think about it, so the main artery that comes off the heart, the heart pumps blood out to, let's say, all body tissues, is the aorta. The aorta then gives off branches of smaller arteries. Those smaller arteries give off branches. Those branches give off branches until you have branches that are just barely visible to the naked eye, and these are referred to as the arterioles. Now, an MVP here, and my background is in sports medicine, so I kind of co-opt that. But in this case, normally we think of MVP as the most valuable player, but in this case, MVP is going to be a most valuable point. So this is a good point to keep in mind, is what I'm saying here. Whenever we take a look at the tunica media, the tunica media of the arterioles is mostly made up of smooth muscle tissues, very little external elastic lamina. And this smooth muscle tissue contracts and relaxes to constrict and dilate the arteriole. So remember, smooth muscle tissue is involuntary. It's controlled by the autonomic nervous system. So the autonomic nervous system will send out the action potentials that will cause the smooth muscle tissue of the tunica media of arterioles to contract. And when it contracts, that would constrict or reduce the size of the lumen of the arteriole, or remove those action potentials, and the muscle will then relax. The smooth muscle of the tunica media will relax, and that will open up the size of the lumen of the arteriole. Now, what this will do is it will regulate the amount of blood that can move into a particular tissue. So if the arteriole constricts, it's going to reduce the amount of blood that's going to flow into the particular tissue that blood vessel supplies. If it relaxes, it's going to increase the amount of blood that flows into that tissue. This results in arterioles helping to do two things. One is to regulate blood pressure. So if the arterioles have the smooth muscle tissue is slightly more contracted, leading to the arteriole being slightly more constricted, it's going to make it more difficult for blood to pass through there, and that's going to increase pressure, the blood pressure. However, on the other hand, if the smooth muscle tissue is slightly more relaxed, the lumen is going to be a little bit more dilated. And if it's a little more dilated, the blood pressure is going to go down. So that's going to help reduce blood pressure. As far as blood shunting is concerned, shunting means delivery of the blood to different areas of the body. So let's say, for example, that I decide I want to go out for a run. And so I'm going to be using the muscles of my legs, and those muscles are going to demand higher amounts of oxygen and nutrients so they can carry out the contractions that are going to propel me down the road as I'm doing my run. So what's going to happen is the autonomic nervous system will dilate arterioles that carry blood into those muscle tissues that are very active, and therefore provide more oxygen, more nutrients, more water for the tissue function. But we only have 5 or 6 liters of blood to circulate through the body at any one time. So if we're going to increase the amount of blood into one tissue, we may have to reduce the amount of blood that's flowing into another tissue. So what's going to happen is, at the time when the autonomic nervous system dilates arterioles carrying blood into the muscles, we may have to constrict the amount of blood, let's say, for example, into tissues of the digestive system, to still provide the digestive tissues with enough blood to supply supply oxygen and nutrients for them to be able to continue to function, but not at a very high rate. So we reduce the amount of blood flowing into the digestive tissues, but we increase the amount of blood flowing into muscle tissues. So this is referred to as blood shunting, delivering greater volumes of blood to the most active tissues and reducing volumes of blood to the least active tissues. And again, the tunica media is very important in that blood shunting process. Okay, let's move from the arteries then into capillaries. And whenever we take a look at capillaries, here's where capillary exchange is going to take place. In other words, substances are going to be exchanged between the blood and the body tissues. And I have an asterisk there. You'll see why in just a minute. Capillaries are microscopic. You can't see them without the aid of a microscope. And capillaries, unlike our arteries and arterioles, are comprised of only a single wall, and that is the tunica intima. So the capillaries comprised of the tunica intima only. The other two walls, the media and the externa, do not exist in capillaries. So that tunica intima consists of that endothelium and the basal lamina. And that endothelium, as I mentioned before, is comprised of simple squamous epithelium that lines the inside of the wall. And here's the reason for that star. Whenever we take a look at the capillaries, we say that there where the exchange between the blood and the body tissues takes place, actually that exchange occurs between the blood and the interstitial fluid. Remember, the capillary is not actually going to touch the tissue. So when substances, and we'll talk about it, capillary exchange a little bit later on in our PowerPoint, but when substances are exchanged, they're exchanged between the capillaries into the tissue fluid, and then from the tissue fluid or the interstitial fluid into the cells of the tissues themselves. So just a point of clarification there. Now, whenever we look at these capillaries, we actually have three major types of capillaries, and we're going to briefly take a look at those. The three types of capillaries are continuous capillaries, fenestrated capillaries, and discontinuous or sinusoidal capillaries. So let's take a look at each of these and see what the differences are between them. First of all, we take a look at continuous capillaries. These are capillaries are the least permeable capillaries in the body and in the tissues. They are very abundant in the skin, in the muscles, in the lungs, and in the central nervous system. Whenever we take a look at continuous capillaries, the reason why they're called continuous is because there are very few gaps between cells. As a matter of fact, the simple squamous cells, instead of having instead of having tight junctions that kind of spot the cells here and there along the membrane, the tight junctions are all along the membrane, and the cells are very tightly adhering to one another. This reduces the amount of oxygen exchange with carbon dioxide and reduces nutrient and waste product exchange. And so, for example, when we talk about the skin, these continuous capillaries will carry warm blood to the surface of the skin, which can then radiate out and through the skin and be released into the external environment to help cool our bodies. But we don't necessarily there want to deliver a lot of oxygen and nutrients via these particular capillaries. Other capillaries that we'll see can do that, but these ones pretty much just allow for radiant release of heat, radiational cooling, we might say. And let's say, for example, in the central nervous system, these continuous capillaries form a major part of the blood-brain barrier that you may recall learning about in A&P1. So the ability to exchange nutrients and respiratory gases, water between the capillary beds and the neurons is greatly reduced to be sure that toxins and microbes that could damage the neurons aren't able to get out of the bloodstream and into the brain tissues. So that forms part of the blood-brain barrier. Okay, our next type of capillary is called a fenestrated capillary. Fenestrated capillaries have large pores in them, or fenestrations, and these help to increase the permeability of the capillary to allow greater osmosis and diffusion of nutrients, osmosis, delivery of water to the tissues, and diffusion, delivery of nutrients to the tissues, and the exchange of those nutrients with waste products. So they're going to have large pores in them. As we can see in these cells, the large pores that there are going to allow easier diffusion and osmosis to occur. We're going to find these types of capillaries in areas of active filtration. Let's say, for example, in the kidneys where the blood is filtered, or areas of active absorption, let's say, for example, in the digestive system, and also in areas where there's going to have to be endocrine hormone secretion, because remember, many hormones are non-steroidal hormones, which means they're basically protein-based. So these hormones are fairly large, so we're going to need these large pores or fenestrations to allow those hormones that are produced in various endocrine glands to enter the capillaries, enter the blood via these capillaries, and then be delivered to other tissues in the body. And then finally, we have those discontinuous or sinusoidal capillaries. So these are the most permeable capillaries in the body, but they only occur in very limited locations. They have very large intercellular clefts and fenestrations. And they have very, very few tight junctions where the cells are actually kind of spot welded together, as we talked about before. So here you can see the fenestrations, and here you can see the wide intercellular clefts that are in the cells. So where are we going to find these types of capillaries? Well, these types of capillaries are going to allow large molecules and even cells to pass across their walls. So we think of places like the liver, which processes a lot of large molecules, and the breakdown products of dead cells. And we think of places like the spleen, where old red blood cells go to be destroyed and broken down. They leave the blood and then pass into the tissues to be torn apart, broken down. This is places where we're going to see these discontinuous or sinusoidal capillaries. They get their name, by the way, from the fact that we can see here the basal lamina. There are even gaps in the basal lamina, so the basal lamina isn't continuous all the way around the endothelium. And sinusoidal, these large clefts are often referred to as sinuses because of their size. And here we go. So we've talked about the liver and the spleen. We'll also see it in bone marrow. In bone marrow, red blood cells are produced, and they have to be able to enter the blood system. So that would be a place we'd find them. And also in the adrenal medulla, where we're going to see large protein-like hormones that have to enter the blood. Okay, continuing with our look at the capillaries then, next up we'll talk a little bit about capillary beds. Capillary beds are networks of many capillaries. Capillary beds are present in all regions of the body. And so, like my fingers, instead of having a single capillary that runs through a tissue, generally what we're going to have are beds of capillaries that run through the tissues and provide blood into those tissues and drain blood from the tissues. So these are referred to as the capillary beds. And within a capillary bed, by the way, we may see, let's say, for example, a continuous capillary and then have some fenestrated capillaries around it. So the continuous capillary allows blood to just pass through the tissue, let's say, for example, in the skin, while the fenestrated capillaries will allow for respiratory gases, nutrient, and water exchange. Prior to the capillary bed, at the end of the arteriole, there are tiny little circular pieces of smooth muscle tissue. These circular pieces of smooth muscle tissue are called sphincters. And since we have these located just prior to the capillary bed, they're called precapillary sphincters. These precapillary sphincters are important. They control blood flow through the capillary bed. So if a precapillary sphincter is closed, no blood's going to be able to flow through that capillary bed. If the precapillary sphincter is open, blood is going to be able to flow through that bed. The more dilated the precapillary sphincter is, the more perfused with blood the capillary bed becomes, and therefore the tissue. The more constricted the precapillary sphincter is, the less blood is going to flow into that capillary bed. So with that in mind, we use the term blood perfusion. So if the capillary beds are open and receiving a lot of blood and allowing a lot of blood to pass into those tissues, we say that those tissues are very perfused with blood. So the precapillary sphincters control the movement of blood into that capillary bed and therefore into the tissues. Therefore, precapillary sphincters control blood perfusion, the movement of blood into the tissues. Okay, next up, we talk a little bit about capillary exchange. Capillary exchange is primarily the result of osmotic and blood pressure. And I go through capillary exchange in our main lecture screencast on that. So I'm not going to go through capillary exchange again. That is in the main screencast, not in this additional screencast that we have on the arteries and capillaries. But I do want to scroll through that very quickly and move on to information on the veins. Now, all that previous information that I went through very quickly is in the original PowerPoint, so you'll pick that up there. I hope this doesn't confuse you, but I do want to get on to the veins because I have adapted some new structure with regard to the veins. Veins transport the blood now toward the heart. So veins are afferent blood vessels. They allow blood to access the heart. Structurally speaking, when we take a look at veins, just like arteries, the walls of the veins consist of 3 distinct layers or tunics. But there are some modifications or differences. The internal layer is referred to as the tunica intima. The middle layer is referred to as the tunica media. However, here's where we have some modification. In veins, the smooth muscle tissue of the tunica media is much thinner than it is in arteries. Therefore, the veins are collapsible. Now, this is in the main PowerPoint as well. And then we have the tunica externa, which is the outermost wall. We also mentioned in the main PowerPoint that larger veins will also have valves in them that permit the blood to flow in only one direction. So they prevent the backflow of blood. Here's the main point I wanted to get to with the veins, and that is this structure. So we wanted this updated structure. Again, we're going to have the tunica intima, and the tunica intima is going to be first of all comprised of that endothelium, and that endothelium is made up of simple squamous epithelial tissue that's going to rest on a meshwork of protein called the basal lamina. So the endothelium and the basal lamina will make up the tunica intima. One of the things you'll notice here with the veins is there's no internal elastic membrane. Veins do not have as much pressure from the blood that they have to resist, so they don't have the internal or external elastic lamina. So we go right from the basal lamina to the smooth muscle tissue. Notice the external elastic lamina is also missing. And then we have the connective tissue, the areolar connective tissue, that then makes up the outermost wall. So whenever we take a look at this, the endothelium and the basal lamina make up the tunica intima. The smooth muscle tissue, and note that the smooth muscle tissue is also much thinner in veins than it is in arteries. That's another reason why veins are collapsible. But the smooth muscle tissue makes up the tunica media, and then the connective tissue makes up the tunica adventitia. And there's our lumen of the blood vessel again, is where the blood actually passes through the internal cavity of the vessel. And there's our note helping you remember this. The internal and external elastic membranes aren't present in veins. Again, that together with the reduced thickness of the smooth muscle tissue means that if veins don't have blood flowing through them and have the blood pressure keeping them inflated, they will collapse if you remove the blood from them. Okay, I think that takes us back to where we pick up in the original PowerPoint, so I'll stop this supplemental PowerPoint here, and you can return to the main PowerPoint or the main screencast on the cardiovascular system 1.